Dear students, hope you are well and going easier. The video on this topic, gastrulation, contain instructive and educational content that is both shareable and easy to retain. So let's start. Gastrulation. Our today's goals are gastrulation, fate maps, gastrulation in sea urchins, gastrulation in amphibians, gastrulation in birds, and gastrulation in mammals. First of all, what is gastrulation? Gastrulation is the process during embryonic development that changes embryo blastula stage with a single layers of cells to gastrula containing the multiple layers. Gastrulation typically involves the blastula folding in upon itself or dividing, which create two layers of cells in diploblastic organisms like jellyfish. While in triploblastic organisms, it creates three layers of cells in higher animals like humans. In diploblastic animals, two germ layers are formed that are ectoderm and endoderm, while in triploblastic organisms, three germ layers are present that are ectoderm, endoderm, and mesoderm. The endoderm will give rise to the gut and associated organs. The ectoderm is the outermost layer and will create the skin and the nervous system. Between them lies the mesoderm, which will create the connective tissue and musculature in most organisms. Fate maps. It is the study in the developmental biology which gives us information about the embryonic origin of various adults and structures. The fate of each cell or group of cells is mapped onto the embryo, showing which part of the embryo will develop into which tissue. We can explain it betterly with the help of the diagram. In the upper 1A diagram, epiblast stage, we can see the three layers that are mesoderm in red color and ectoderm in blue, while endoderm is in yellow color. When these structures are developed, they give blue color will give rise to the blue, and red one will develop into the mesoderm, and yellow will develop into the endoderm, from which we can say that it gives us information about that which part of the embryo will develop into the which tissue or organ or structures. Gastrulation in sea urchins. It involves two separable morphogenetic processes. Number 1, ingression of primary mesenchyme cells. Number 2, invagination of the archenteron. This is a general overview of the gastrulation in sea urchin. Sea urchin gastrulation begins with the ingression of primary mesenchyme cells epithelium at the vegetal pole of the embryo, known as the vegetal plate. Following ingression of primary mesenchyme cell, there is a pause and the vegetal plate invagination to form a stout cylinder called the archenteron. After another pause, secondary mesenchyme cells appear at the tip of the archenteron, and at about this time, the archenteron begins to elongate across the blastocoel. Eventually, the tip of the archenteron makes contact with the ectoderm near the animal pole. Meanwhile, the primary mesenchyme cells form patterned array and they ultimately secrete calcium carbonate containing skeletal rolls called spicules. The ventral ectoderm flattens give rise to what is known as the prism stage. Finally, the gut ectoderm and skeletal undergoes the further obvious differentiation to make the Plutus larva. Ingression of the primary mesenchyme cells. In this, primary mesenchyme cells undergo ingression at the onset of gastrulation. Although the mechanism of ingression are not fully understood, several events are known to accompany ingression. The best characterized changes in the primary mesenchyme cells involve changes in their adhesive properties. In these steps, the vegetal plate undergoes invagination to produce the archenteron. Since sea urchins are deuterostomes, the plastopore form the anus of the later larva in development. The vegetal plate is an epithelial monolayer, and so the process of primary invagination seems like a desperately simple case of invagination. However, appearances can be deceiving. The mechanism of primary invagination are not well understood. However, several recent experiments indicate at least some of the requirements of primary invagination to occur normally. Number 2 is ingression of the archenteron. Secondary invaginations involves the elongation of the archenteron across the blastocoel, where it attaches to the ectoderm near the animal pole of the embryo. The onset of the secondary invagination correlates with the appearance of long, thin philopodia extended by the secondary mesenchyme cells at the tip of the archenteron. Under a variety of circumstances, there was a substantial evidence that secondary mesenchyme cells were required for extensions of the archenteron during secondary invagination. Then what is philopodia? Secondary invagination is thought to involve philopodia extended by the secondary mesenchyme cells located at the tip of the archenteron. The onset of the secondary invagination correlates with the appearance of long, thin philopodia extended by the secondary mesenchyme cells at the tip of the archenteron. Under a variety of circumstances, there was substantial evidence that the secondary mesenchyme cells were required for the extension of the archenteron during secondary invagination. Convergent extensions. Secondary invagination also involves one thing that is called convergent extension. In this convergent extension, how does the archenteron elongate? There are some questions that how does the archenteron elongate given that both autonomous extension and secondary mesenchyme cells dependent extension occur. So there is sometimes scientists show that sea urchin gastrulation elongate their archenterons via a convergent extension. A more direct demonstration of this was provided by Hardin in 1989, who used fluorescent clones of cells in the chimeric embryo to show that epithelial cells of the archenteron rearrange as it elongates. Note that on the left the clone has smooth boundaries. By the end of the gastrulation, the clone has intercalated with the neighboring unlabeled cells to generate a jagged boundary. So here we can see in the diagram in the right side and in the left side. In the left side, clones has smooth boundaries, but by the end of the gastrulation, in the right one diagram, the clone has intercalated with the neighboring unlabeled cells to generate a jagged boundary. This was all about the sea urchin. Here is the flowchart diagram of sea urchin. There are some steps described from which we can understand best that which steps are involved in the gastrulation of the sea urchin. In number 1 step, some blastoderm cells invaginate to form the archenteron. In number 2, other blastoderm cells ingress becoming primary mesenchyme. In number 3 step, cells along the sides of the archenteron elongate. Number 4, the mouth will form where the archenteron meets with the ectoderm. Number 5, mesenchyme cells will form the mesoderm, and the blastopore will form the anus of the mature animal. Next is gastrulation in amphibians. We have already discussed what is fate mapping. Fate mapping of blastula stage embryo has provided some insight. Number 1, using vital dyes to mark cells. Number 2, superficial layers of embryo form the ecto and mesoderm. Number 3, mesoderm lie mostly in the deeper layer of the cells. This is a diagram of exterior and the anterior view of the gastrulation in amphibians. In an exterior side, epidermal, suprablastoporal endoderm and subblastoporal endoderm is formed, while in the anterior side heart, somites and notochords and lateral plate of mesoderm is developed. In the exterior, neural plate was developed. Cell movement in amphibian gastrulation. Gastrulation begins on the dorsal side below the equator in the region of the gray crescents. Cell migration to form a cylinder blastopore. Then what is sapmen organizer? Dorsal lip of blastopore will become imported organizer region of the embryo, which is called the sapmen organizer. Then what is bottle cells? Cells become elongated as they contact the inner surface, that is called the bottle cells. Here is the diagram of the movement of the cell. We can see in the vegetal pole and in the animal pole as well. Bottle cells are present in the inner side. We can see in the diagram in the yellow shade we can see the bottle cells and the dorsal blastopore lip. While in the animal pole, blastocoel is present and in the superficial cells are also present. Overview of amphibian gastrulation. Here is the overview of the amphibian gastrulation, from which we can understand the steps involved in the gastrulation of amphibian. In number 1 step, gastrulation begins when cells just below the center of the gray crescent invaginate to form the dorsal lip of the future blastopore. Cells of the number 2, cells of the animal pole spread out, pushing surface cells below then toward across the dorsal lip. Those cells involved into the interior of the embryo, where they form the ectoderm and endoderm and the mesoderm. Number 3, this involution creates the archentron and destroys the blastocele. The dorsal lip form a circle, thus which cells moving to the interior are around the blastopore. The yolk plug is visible through the blastopore. We can see in the diagram as well, there is all cells are labeled, we can understand it betterly. In the number 4 step, continued development, which give rise to the notochord derived from the mesoderm, and in the last one, the beginnings of the nervous system, which show in the green color, are derived into the from ectoderm. This was about the gastrulation in amphibians. Gastrulation in birds. Egg laying occur in blastula stage. It is followed by the incubation at 38 to 40 degrees Celsius. Due to the large amount of yolk, gastrulation is modified and prolonged. It starts about 4 to 5 hours after laying and completed by the second day of incubation. Process of gastrulation. Number 1, formation of the endoderm. Number 2, formation of primitive stick. Number 3, formation of mesoderm. Number 4, formation of notochord, and the 5 one is the formation of mesoblastic somites. Number 1 is the formation of the endoderm. We can see in the diagram as the formation of the endoderm starts when the endoderm of the hypoblast developed as a single layer of cells inside of the blastocele. After the formation of endoderm, upper layer is called the epiblast. There are different forms, we can say, in the endoderm of hypoblast. Formation of the primitive streak. The second step in the gastrulation is the formation of the primitive stick at the posterior region of the area pellucida in the mid-dorsal line. Primitive stick will appear as the thickened area. It starts 8 hours after incubation. The thickening is because convergence of cells of blastoderm towards the center. Usually in the earlier stages, the primitive stick is short and broad. It gradually extends to the middle of blastoderm. At 18 to 19 hours of incubation, primitive stick is well developed. It is called the definite primitive stick. The primitive stick has a primitive groove, 2 primitive folds, a Henson nodes, a primitive pit, and a primitive plate. This is the primitive stick convergence, invagination, and divergence. We can see here the diagram. There are convergence, invagination, and divergence. Arrows shows that in convergence states, this enters in the primitive stick and invaginate, and then they diverge from the primitive stick. These 3 types of movements during the formation of primitive stick occurs, that is convergence, invagination, and the divergence. Formation of the mesoderm. The mesoderm is formed as 2 layers. In the front of the primitive stick, an area without mesoderm is present. It is called the promonion. At this place, head will develop. After 48 hours of incubation, the promonion is also occupied by the mesoderm. The mesoderm is divided into dorsal and intermediate and lateral mesoderms. Then, in the next step, we can find the notochord formation. Formation of the notochord. The notochord cells arrange themselves to form a slender rod called notochordal process. It will begin at Hansen nodes and it slowly grows. Because of its growth, the primitive cheek is slowly reduced. By the end of the gastrulation, the primitive cheek is reduced and incorporated into tail bud. The dorsal mesoderm is located on the either side of the notochord. It is divided into segments that are called somites. Formation of the mesoblastic somites. The dorsal mesoderm is located on either side of the notochord. It is divided into segments that are called somites. The first pair of somites will form after 21 hours of incubation. The blastoderm develops a mid-dorsal thickened area called the primitive cheeks. Gastrulation is completed within 2 or 3 hours after laying. This is the diagram of chick's egg. With its completion, the embryo becomes diploblastic, containing of an outer epiblast, ectoderm, and an inner hypoblast, endoderm, and the original subgerminal cavity is divided by the hypoblast into a narrow outer space, or secondary notochord, and inner archantron or primitive gut of embryo. All about the gastrulation in birds. Chick embryo is described in the form of flowchart diagram. There are some steps from which we can understand betterly that what steps are involved in the gastrulation of birds. In number 1 step, the posterior epiblast thickens. Number 2, cells move towards the primitive cheek, down through it and forward. Number 3, the primitive cheek narrows and lengthens. Number 4, forming the primitive group, the cheek's blastopores. Cells passing our Hansen node form the head structures. Number 6, these cells will form the mesoderm. These will become endoderm. We can see in the diagram as labeled, and flowchart is described how the gastrulation occurs in the birds as well in the chick embryos. Gastrulation in mammals. During gastrulation in mammals, epiblast cells converge at the midline and ingress at the primitive cheek. Ingression of these cells result in the formation of the mesoderm and replacement of some of the hypoblast cells, produce the definite endoderm. At initial, as gastrulation proceeds, the primitive group extends anteriorly. Germ cells. A cross-section through the embryo allows us to observe 3 germ layers, that are ectoderm, endoderm, and mesoderm. The most characteristic event occurring during the 3rd week of gastrulation is the gastrulation, the process that establishes all 3 germ layers in the embryo. Gastrulation begins with the formation of the primitive cheek on the surface of the epiblast. Initially, the cheek is vaguely defined, but in a 15 to 16 day embryo, it is clearly visible as a narrow groove with slightly bulging regions on the either side. Primitive cheek of the epiblast. The cephalic end of the streak, the primitive node, consists of a slightly elevated area surrounding the small primitive pit. Cells of the epiblast migrate towards the primitive cheek. Upon arrival in the region of the streak, they become flask-shaped, detached from the epiblast, and slip beneath. This inward movement is known as the invagination. Human embryo at 9 days. We can see here the embryo at 9 days. We can see the wall of uterus, development of placenta. This inward movement is known as invagination. We find in the last slide. Cells migration and specification are controlled by a fibroblast growth factor, which is synthesized by the streak cells themselves. These growth factors control cell movement by down-regulating a protein that normally binds epiblast cells together. Fibroblast growth factor then controls cell specification into the mesoderm by regulating expressions. Once the cells have invaginated, some displace the hypoblast, creating the embryonic endoderm, and others come to lie between the epiblast and newly created endoderm to form the mesoderms. Cells remaining in the epiblast form the ectoderm. Thus, the epiblast, through the process of gastrulation, is the source of all the germ layers. And cells in three layers will give rise to all of the tissues and organs in the embryo. Human gastrulation 13 to 19 days. As more and more cells move between the epiblast and hypoblast, they begin to spread laterally and cranially. Gradually, they migrate beyond the membrane of the disc and establish contact with the extra-embryonic mesoderm, covering the yolk sac and amnion. In the cephalic direction, they pass on each side of the precaudal plate. The precaudal plate itself formed between the tip of the notochord and oropharyngeal membrane and is derived from some of the first cells that migrate through the node in the midline and move on the cephalic direction. Later, the precaudal plate will be important for the induction of the forebrain. The oropharyngeal membrane at the cranial end of the disc consists of small region of the tightly adherent ectoderm and endoderm cells that represent the future opening of the oral cavity. So here is the embryo. At the first diagram we can see the actual size, that is 0.2 millimeter, and in the number 2 diagram actual size is 0.4 millimeter, while in the 3-1 diagram we can see the actual size is 1 to 0.1 to 0 or 0 to 1.5 millimeter. This was all about the gastrulation in human. This was a short overview of gastrulation. While gastrulation in human period was too long to describe, this was all about the gastrulation. Thank you to all of you.